matters. The topic for today is base editing and decreases in LDL cholesterol and cardiovascular disease. This recent information is part of a rapidly changing landscape in the treatment of cardiovascular-related problems, what many are calling game-changing science and technology. I think it's actually catching up, Dr. Gonshor, so you roll it.
Well, Reed, good to see you. And as we always say in each one of these sessions, we take a deep dive into recent research. And the key is recent. We want to make sure that you know what's going on right now to bring to light objective scientific evidence for whatever the topic of hand is. And today we're going to be doing things a little differently, and you'll see why. We will not be looking at a published study, but rather at a presentation that took place at the American Heart Association on November the 12th. So it's literally just a few days ago. This presentation, together with a large number of lead-up research studies, is really beginning to show solid evidence of the significant role that's being played by CRISPR, and we'll discuss what that is, and RNA therapy. So let's look more closely at base editing and decreases in LDL-C and cardiovascular disease, because science matters.
Yeah, that's fantastic. So I like the fact that this study, Dr. Gonshor, was more of a presentation than the typical studies we read. I like the variety there. So, but before we get into that, I just want to say on our previous broadcast, which was antioxidants stimulate BOC1-dependent tumor angiogenesis, that that stimulated a number of questions. So I hope it's okay if I ask you one of them. It's kind of one of the big ones, which was that where the research stands today on the inhibition of angiogenesis.
Well, it's a good question. It's an important question. Angiogenesis, which is the creation of blood vessels, plays a very important role, not only in the development of cancer, but in metastasis, in cancers moving out from their primary location to the rest of the body. To do that, they need blood. They need blood vessels and the blood that comes with it, since it really allows for the delivery of oxygen, of nutrients, growth factors. And so that's the way that a tumor can disseminate to distant organs, something you don't want when you have malignant tumors. So inhibition of angiogenesis clearly is an important strategy for the prevention of multiple types of solid tumors. And it really depends on cutting out completely, or at least reducing the blood supply to a tumor. If you can starve that tumor of oxygen and nutrients by not allowing those blood vessels to form, then you have a chance to be able to really be in a good fight against that tumor. So the inhibitory effect can be achieved. In this case, we're discussing angiogenesis. So it can be achieved by inhibiting the expression of various angiogenic factors in the tumor. What does that mean? Factors such as vascular endothelial growth factor. Many of you know that as VEGF, which is a very potent angiogenic factor, or basic fibroblastic growth factor. That's another potent angiogenic factor. What does that mean? These are particular entities that create the stimulus for blood vessels to form. And so that's what you're really trying to inhibit. So angiogenesis inhibitors work in a special way to combat cancer, which do not explicitly kill the cancer cells themselves, but it's targeting the signals which really support the tumor's blood supply. Again, cut out the blood supply, the oxygen, the nutrients, you stand a good chance of really killing or inhibiting those cancer cells' ability to work. So several of these drugs work by binding directly with, for example, VEGF, blocking it from doing what? From sending signals that will stimulate angiogenesis, that will stimulate those blood vessels to form. And in that way, it maintains a balance between activators and inhibitors, which is essential for ensuring vascular homeostasis. So just to make sure you understand, as a standalone therapy, inhibition of tumor angiogenesis, it can arrest or it can halt tumor growth, but it won't eliminate the tumor. So when you're doing therapies in which you're using anti-angiogenic drugs, you need to do that in combination with other anti-cancer treatment methodologies like chemotherapy, etc. And that is critical for optimum cancer patient outcomes. By the way, this is changing by the day. So I want to make sure you understand this kind of information literally is a weekly or daily, in some cases, change. We're in a very wonderful period of changes in technologically driven therapy.
Well, this therapy to me looked safer, and we've run a lot of VEGF testing on people if they had kind of a chronic inflammatory response kind of a thing. So really interesting there. And that recording, by the way, it's a great recording, a really good presentation by Dr. Gonshor, and you can go back into our archives and watch that. I want to thank you guys for submitting your questions, and they're all important as we learn together, of course. So thank you, Dr. Gonshor. Let's jump into today's study about base editing and decreases in LDL cholesterol and cardiovascular disease.
Well, this is a fascinating thing to be looking at. And we're talking base editing and decreases in LDL-C and cardiovascular disease. We'll describe what that is. And as I said, this is not a paper, but it was a presentation at the American Heart Association just a couple of weeks ago, and it is a blockbuster. So it was really discussed as a proof of concept. And the idea was, in the words of Dr. Bellinger, who said the following, this is a technique. It's a proof of concept of a technique for precisely rewriting the genetic code directly in the body, and how that can slash what we call bad cholesterol levels, possibly for life. Remember that for a moment, possibly for life. And in this case, it was in 3 people prone to dangerously high levels of this artery clogging fat. And the feat relied on blood infusion of a so-called base editing therapy, and we'll go into this in detail, designed to disable a liver protein, PCSK9, that regulates cholesterol. So that was the statement made by this Dr. Bellinger of Verve Therapeutics. And it needs to be understood how important this is. This is the first time that CRISPR, and we'll describe what that is, a CRISPR variant has been infused into people to treat a disease. And the success is also a proof of a principle for using gene editing for a common health problem like high cholesterol, which is a major problem worldwide. And that's important because they're doing it for cholesterol rather than various rare diseases. And this base editor that he discusses could eventually be a one-time solution. Think about this, a one-time solution for tens of millions of middle-aged people who are struggling to control their cholesterol with daily drugs. So what is this CRISPR-Cas9? Well, it's an acronym. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. You can see why they call it CRISPR and not this long name. And what is it? Well, CRISPR is actually a family of DNA sequences in the genomes of organisms like bacteria or archaea. These are all called prokaryotes. Bacteria, when they get infected by viruses, they actually attack the virus, and the part that has attacked them, that area of DNA that they used, they keep that piece of DNA, and that's that clustered, regularly interspaced, short palindromic repeat, the CRISPR. It keeps it around so that it knows that if the virus tries to come in again, it's already got a piece of knowledge that has remained, almost like an antibody that is going to be able to attack that virus. So several years ago, around 2012 and 13, the first papers came out on CRISPR technology. The two people who published papers on it, Jennifer Doudna out of Berkeley, and I can't remember the one from France, Dr. Carpentier, they won the Nobel Prize about 2 years ago, probably one of the greatest discoveries of the last 100 years. And the idea is that you can use this particular kind of technology based on what they learned that bacteria are using to be able to then deal with DNA by using, in effect, a genetic engineering tool, using this CRISPR sequence in the DNA, and to edit base pairs of a gene, to cut out the parts that you don't want, and to insert right into that place the snippet of a gene that is normalized and that you really want to have in there. So CRISPR severs both of the strands of DNA and in effect lets the cells to a great degree repair themselves. But this can result in some cases in potentially harmful DNA rearrangements that could flip on a cancer gene or something like that. Base editors is a bit of a twist on CRISPR. It's part of a CRISPR technology, but a little more precise, because what it does, it cuts out just one DNA strand and it swaps out pairs of the 4 DNA bases. And we're going to go into that in just a moment. It was first clinically tested, this is the base editing type of CRISPR, in 2022. That's a year ago. The researchers at that point had engineered donated immune cells. They put them in a dish, and they were going to target a patient with leukemia. So what they did is they rearranged those cells in the dish using CRISPR. They then infused these cells that had been changed to put the disease into remission. And that allowed time to get a stem cell transplant and so on. In this trial by Verve Therapeutics that was just dealt with a couple of weeks ago, the editing took place not in a dish, but right inside the body, specifically in the liver, which is a relatively easy organ to target because we know that it sucks up foreign particles. That's what it does for a living. And so how did they do this? Well, they used the same technology which was used for mRNA, for vaccines, and so on. What they used were little lipid nanoparticles. And I'm giving you just a picture of some of these nanoparticles. I hope you can see them. This is on electron microscopy. They look something like this, nanoparticles, which are incredibly small. And inside them, and the nanoparticles are the delivery system, because inside these little particles is the RNA therapeutics. Why do you need the nanoparticles? Because if you didn't have some kind of a coating over these RNA substances, the RNA would be degraded very, very quickly in the intestine when you put it in vivo. So this lipid layer not only encapsulates the mRNA, but also it's able to fuse with a cell membrane, as I show you here on the schematic. And as soon as it fuses, it allows the RNA that was encapsulated in these nanoparticles to enter the cell and then to do whatever it needs to from a therapeutic standpoint. So in this clinical trial, the subjects that were chosen have a disease which is called heterozygous familial hypercholesterolemia. Big word. It's basically very, very high levels of cholesterol, and it's caused by a defect in one copy, just one copy of a gene that encodes what? It encodes a cell surface protein receptor that is needed by the liver to clear the blood of LDL. That's the low type of lipid. This defect results in the receptor not being around long enough or in sufficient amounts to keep this LDL level in check. So normally when the receptor is working well, sticks around for a while, and what it's able to do is to clear the LDL from the body. But if it doesn't stick around long enough, that LDL builds up. So the strategy of the study, keep those surface receptors around longer. And how do you do that? You do that, in their case, by altering a liver's gene, a gene called PCSK9, which is an enzyme. It's a gene for an enzyme that normally removes these receptors from the cell surfaces. So the idea is, if you can stop that enzyme from working, those receptors don't get broken down by the enzyme, so they stick around longer. If they stick around longer, they're then going to allow the liver to clear the blood of LDL. So in the treatment, the messenger RNA instructs cells to manufacture the gene editor's protein components, because remember, they're packaged in these little lipid nanoparticles. So they travel to the liver, these particles do, where there's an additional RNA strand also carried in the particles that guides the base editor to this gene for that enzyme. So in the nanoparticle, there are two types of RNA. One RNA is what we would call a guide RNA. It says, go over there. And the second strand is the one that's going to do the cutting of the DNA. So this combo makes a one base pair change, just one base, so that cells can produce what? Only shortened, non-functional versions of the enzyme. And if that enzyme is not functioning, it results in the receptor remaining on the cell surface. So this company, Verve Technology, with its Verve 101, what was it able to do? It says it's a single course treatment designed for those people with this particular of cholesterol, the HeFH group, and it does it by inactivating this enzyme's gene in the liver to durably lower blood LDL. So what they did is they made a change in adenosine to guanine, just a change of two bases in the DNA genetic sequence of that enzyme, and it was able to inactivate that target gene. Inactivation of that gene for that enzyme had been shown to allow receptors to stick around. So it upregulated, meaning it allowed an increased level of that LDL receptor to remain on the surface, leading to lower LDL levels. Because remember, you need that receptor around to help the liver decrease LDL. And by doing that, it was able to reduce the risk for atherosclerotic CVD or vascular disease. And this HeFH is just a subtype of that kind of vascular disease. So the results, and this is interesting, following a single infusion of this particular treatment, patients showed a dose-dependent reduction in the blood levels of that enzyme, an outcome that suggests successful editing at the intended genomic target. 1 month after the treatment with this particular substance, these patients showed a dose-dependent reduction of disease-causing low-density lipoprotein cholesterol, which is wonderful. And so the clinical implication is actually quite mind-boggling. It could open the way for an entirely new way to treat heart disease. As they quoted, instead of daily pills or intermittent injections, you'll get a one-time IV infusion procedure to durably lower LDL cholesterol. In fact, that may last lifelong. Verve, this company, plans to test this treatment in a total of 40 of these type of patients. The current participants were only in New Zealand and the United Kingdom. The US regulators literally just a few weeks ago cleared the way for testing. So it expects to compare the approach to a placebo and a typical objective study in a much larger trial starting in 2025. And Verve Therapeutics, this company, says that this enzyme base editor could one day be used to treat people who don't have this particular subtype, but they have early heart disease. So it could even be, it could be given widely to older adults to ward off this disease, cardiovascular disease. The authors note, in fact, that, quote, down the road, maybe when you turn 50, this is what you would get, and it would prolong your life. And that's the ultimate vision. That's normally not what you see from scientists. So what's the key takeaway? There's a technique for precisely rewriting the genetic code using base editing therapy that holds the possibility of slashing bad cholesterol levels, the LDL, possibly for life. Gene editing with this CRISPR format enters important new territory, which may lead to future treatment for and ultimately a vaccine against heart disease. So the door is now wide open for precise corrective editing to definitively treat grievous genetic diseases centered in the liver, including inborn errors of metabolism. I get shivers just thinking about this. And that's it for today's Science Matters.
Fascinating presentation, and you explained some things. It's really an important area for us genes. You know, we have an epigenetic program, and here it looks like the ultimate shortcut, you know. But as to could it lead to something besides vaccines, that because our population won't take them, they don't trust them, they don't want anybody sticking anything in them that they don't understand. And is there another way to use CRISPR to develop something else? You know, that technique of reprogramming the gene, that could be valuable for sure. But if it's only just another way of producing a drug that's probably going to be very, very expensive to change the genes, which probably is better than taking an anti-cholesterol medication for years and years and years. You know, yeah, if you could change the gene and not have to take all that medication, that seems like a valuable pursuit. I'm just wondering what the auspices around that therapy would be, because if it could turn into some kind of a treatment, fine. But if it turns into a vaccine, that's a whole different ballgame. You just opened up a new can of worms about this whole thing.
You need to be careful. Sure, vaccines can be something that you get with mRNA base editing, but this, let's not confuse it. And I hope I didn't confuse people. Vaccines is one thing, but here you're looking at non-vaccine base editing. You're literally changing, you're cutting and putting in the right base where in fact it was wrong. That has nothing to do with vaccines, although you can get vaccines for various things. But this is a clear, very precise, even more precise than the precise CRISPR. They're getting down to literally saying, you see where that adenosine is? I want to change it to a guanine. And they literally do that. That's it. And that changes the ball game completely.
Well, thanks for that clarification. So I get it, and I'm learning, and we're working more use of gene testing into some of what we do in FDN. Well, thanks for those takeaways. So to our audience, we'd like to say thank you for being here. I want you to subscribe, like, and share if you enjoyed this episode. And as always, you can submit your questions, comments, suggested topics at www.fdntraining.com/science matters. And if you want to grab your own copy of the studies, go to the same place, www.fdntraining.com/studies. And so join us again, please, in 2 weeks, December 27th, when we'll talk about the gut microbiome, mild cognitive impairment, and probiotics. It's a really great study, and I want to thank you very much because
science matters.
matters.